It's a pleasure and an honor to be here and given the opportunity to address you folks. So I thought of giving a slightly different type of lecture about MS than you probably are used to. I'm not an immunologist, I'm not even an MS physician, I'm a stroke doctor, but I come into the MS field a number of years ago with a very different view of what might cause damage to white matter and axons. And I guess that's an advantage in one way, disadvantage in another, but it allowed us to start with some new thinking about what might be at the root cause of the disease. And I'll give you my personal thoughts about what this disease might be all about, because as the title implies, we really don't know the answer, but we're going to look at it through this grant and the generosity of the society in a slightly different way to maybe get some new insights into this disease. So my talk will be structured in basically 4 parts. I'll give you a brief introduction about what the traditional view of MS is, the nerve axons, as Gary mentioned, the white matter of the brain, which is the main but not the only target in MS. Then we'll go on to what I call the fly in the ointment, kind of inconvenient, if not truths, inconvenient observations about the disease that kind of make me rather uncomfortable about the conventional thinking of MS. I'll show you a little bit about how we approach the disease in our lab, the tools that we use that you might find interesting, and finally a couple of slides wrapping up what I think might be the real disease and where research might want to focus more. So if you Google around MS, and I'm sure you've all done this, you come up with a traditional definition of MS. This is what we were taught in medical school. It's an inflammatory disease. There's no disputing that. Inflammatory cells, lesions on MRI, markers in the cerebrospinal fluid of MS patients all point to a highly inflammatory autoimmune disorder. There's no disputing that at all. That's a fact. You see the myelin mentioned in definitions of MS. Myelin is that fatty wrapping of nerve axons in the brain and the spinal cord that is absolutely essential to allow transmission of nerve impulses, and one which is the main target of damage in multiple sclerosis, causing symptoms and signs in our patients. Inflammation in the immune system is a very common theme, of course, because it is an immune disease. It is an autoimmune disease. There's no disputing that. The question in my mind is that the primary problem? And of course, this is still true after 150 years of recognition of the disease by Charcot back in the 1800s, and after a good many decades of very intensive investigation by the MS research community, we still don't know what causes it. So white matter, as you might know, makes up 50% of the brain. Here's an MRI scan, a coronal scan, so if you cut the brain like this and looked at the brain. The gray matter, the cortex, is on the outside. The deep inside, the white stuff here on MR is the white matter. That's where all the nerve axons and myelin live and serve the vital function of connecting neurons to various parts of the brain and the body. So the brain is divided into gray matter, where the nerve cell bodies and their trees live. The synapses are here. And each neuron has a single axon, its connection to the world, if you will. And this is where the problem is in multiple sclerosis. So I like this cartoon, and this is kind of the one I use when I used to give stroke talks a lot when I did stroke research, that most of the world works on the nerve cells, but very few researchers work on the white matter axons, and this is where we have focused on, because to me, if you have a telephone and you can't plug it into the wall, you can't connect it to anything, it's not of much use to you. So this is kind of my raison d'etre for my entire career. And axons have a particular problem. They are long, they're tiny, and they have to connect, and if they're interrupted at any point on their course, they're out of the picture. So if you take an average meter-long, 4-micron diameter axon in the spinal cord of an adult human, and you stretched it and you expanded it to the width of a 4-lane highway, that single axon would stretch all the way from one coast to the other of our vast country. So that is incredible, and that axon has to survive, be healthy, and function properly for the 80-plus year lifespan of an average human. And that is a remarkable feat, because at any point of interruption along that point, the axon ceases to play its role, ceases to function. So axons are absolutely remarkable devices. They're serial devices because they have to work serially from beginning to end. And this is what intrigued me way back when I was in medical school, when I decided this is what I want to work on. So white matter is affected by many diseases. As neurologists, we see lots of terrible diseases that affect patients, not only MS. There's a stroke patient of mine, a devastating stroke. Even tiny strokes cause big problems. Here's a small stroke in a critical part of the brain. The internal capsule, which is all white matter, and that patient is completely paralyzed on one side. Because of this, traumatic brain injury affects white matter in a microscopic diffuse manner. Spinal cord injury, of course, is a terrible situation where a very small volume of interruption of mainly white matter axons causes quadriplegia, a terrible situation that is irreversible for the patient. And the disease of most interest to us today is MS, where something goes wrong with the white matter, not only white matter, but mainly the white matter. So MS, unlike the other examples, is a dynamic disease. I'm glad, David, thank you for reminding me that my video wasn't loaded, so it is loaded and working. And this shows you that this is a scan of a single patient taken at 1 weekly intervals for a year. And it shows you the dynamic nature of MS. All these white spots are inflammatory lesions that come and go, mainly in the white matter of this patient. Every week, lesions come and go, come and go. And that does not correlate with disability. This patient had very few clinical symptoms, underlying the fact that lesions are coming and going, probably causing damage in their wake, but clinically, the patient or the clinician does not necessarily see the dynamic nature of MS unless you study it in this fashion. So very dynamic, very active, something is going wrong all the time that we're not aware of. The problem, though, after these lesions burn themselves out with time, is that many, in fact most patients, despite treatment, we'll get to that later, progress to atrophy. So under the background, under the hood of this dynamism, if you will, the brains of MS patients gradually atrophy. MS patients lose neurons, they lose axons, and they lose brain volume. And that is the single biggest problem that we have no treatment for, and that we believe underlies the progressive, irreversible disability that most MS patients suffer, unfortunately. So what's going on microscopically? Well, we knew that for a long time. Back in the 1800s, Charcot took brains of MS patients, looked at them under the microscope, and found myelin loss, called demyelination, found swellings and transections of axons, of the nerve fibers. And although demyelination is somewhat reversible, our brains have a certain capacity of repairing the stripped myelin. Once an axon is cut, it is cut permanently. Unlike the peripheral nervous system, where if you have a nerve injury in your hand or your arm, you may get some function back. Once you injure a nerve axon in the brain or spinal cord, it's gone. Luckily, our brains have lots of redundancy. We have billions of nerve fibers, so we can withstand a lot of loss. But sooner or later, when you lose enough of these fibers, as shown by Bruce Trapp a decade or so ago, very nicely quantitated this. And all these swellings here, this is from a brain of an MS patient. These are axons that have swollen up and have gotten cut. And once they swell up and get cut, they no longer transmit. And as I showed you on that slide, with the highway in Canada, when they don't transmit, then that axon is taken out of the picture. A very common pathological hallmark of MS is inflammation, of course. And here's a classical example of all these dark blue cells. These are all inflammatory cells that are clustered around a blood vessel, typically a small vein in the brain of MS patients. And when these guys come in, are recruited into the brain, they wreak havoc. They secrete lots of nasty molecules that propagate the damage and cause even more damage to the myelin and the axons. So there's no question that MS brains are full of this. The question in my mind is this a reactive thing, or did this start the whole process? I'll show you some arguments later. So here is a cartoon that basically summarizes the two key problems that we're faced with in the MS research community. That is first, loss of myelin. So here's an axon with normal myelin sheath that's interrupted at regular intervals, called nodes of Granville, which is perfectly normal. But at some point, and for some reason, this myelin starts to break down. And then later, once the myelin breaks down, the axon swells up and gets cut. This is partially repairable. This is totally irreparable. And this is the main underlying problem that we're trying to understand and fix in MS. So inflammation, gadolinium-enhancing lesions on MRI, markers of autoimmune reaction in the spinal fluid of MS patients, those are all very prominent things that MS clinicians see in their patients. Those are things that prompted decades of intensive investigation to develop very effective drugs to combat this inflammatory response. And if you were sitting in the Pacific Ocean and looked at a nuclear detonation in the Bikini Atoll, what you would see from 20 kilometers away is the huge mushroom cloud. That is the obvious thing, the obvious reaction to the detonation of a refrigerator-sized device that was sitting at the surface. So it is completely natural for the research community to to have gone after the mushroom cloud. We go after the autoimmunity, obvious inflammation that we see in MS. And we've got really good drugs for that. But that doesn't mean that the cause of the mushroom cloud was not some small device sitting on at ground zero that was totally obscured by that mushroom cloud. So we're taking a different view. We're saying, okay, we've learned a lot about the autoimmunity and the inflammation. And even after learning a lot, we have no cure. We have no cause for MS. So we look at it differently and say, well, wait a minute. Maybe something caused the mushroom cloud. Maybe there's something down here on the beach that we need to look at, and that's our approach. So this cartoon kind of summarizes that approach. So the main focus of MS research has been autoimmunity, inflammation, and there's also degeneration that has been recognized. But everyone assumed that this starts first, and then in turn causes damage to cause later degeneration. We're thinking maybe it's backwards. Maybe there's something that started here, and for some reason the MS patient loves to react to degenerated white matter. And secondarily, there's lots of inflammation that comes in, and itself causes lots of damage. So it's a real chicken and egg argument, and the bottom line is we do not have an answer of which comes first. But what I'm saying is that let's not assume, because the mushroom cloud is so prominent, that the answer must lie where the most prominent phenotype or prominent manifestations are. So MS is an inflammatory disease, there's no question. Here's the fly in the ointment thing. So when I started looking at this disease in detail, you look in the literature and you see interesting things from other diseases that might teach you. So starting with MS first, one of the flies in the ointment for me were publications like this one from John Prinius' group in Australia. So what he did is he decided to look the occasional unfortunate patient who had a very acute attack of MS and for whatever reason passed away very quickly after the acute attack. And what Prinius and colleagues did is they got the tissue, got the brains of these patients, and when they looked at them carefully, this is what they found. So this is the brain, this is the cortex, the gray matter here, that's normal. All this blue stuff is a stain called LFB, and that stains myelin, all that rich fatty stuff, and white matter of the brain. And this whole area should be blue, and in this patient, this area is very, very pale, and that means the myelin was broken down, was stripped off, was taken away. So all these nerve fibers are demyelinated, they don't work. This is a huge MS plaque that is undoubtedly causing this patient lots of disability. But here's the fly in the ointment. If MS was an autoimmune inflammatory disorder, what these investigators did not find is tons of inflammation in this plaque. There is in fact no inflammation in this plaque. There's no antibody deposition in this plaque. So if this huge plaque was caused by an inflammatory autoimmune process, where is the inflammatory cells? Where are the antibodies? They're not there. So when you see things like that, it makes you wonder, did this start first? And if this patient had survived and you waited long enough, then you'd see autoimmune cells coming in and inflammation starting. So that's a very curious thing in my mind. Here's another example. So nerve axons, the myelinated fibers I showed you about, if you take them and cut them in cross-section, look at them under EM, you see something like this. This is a beautiful structure in my mind. So the axon itself, the actual copper wire is in the middle, cut transversely, and around it you've got these beautiful wraps of highly regular lipid-rich myelin, the myelin sheath, and that insulates the axon electrically. So Rodriguez and colleagues at the Mayo Clinic, they did a lot of biopsies on acute lesions that turned out to be MS later, but at the time they didn't know that, so the neurosurgeons went in to try to make a diagnosis and did biopsies. So after the fact, they figured out this is MS. But very early on, they saw pathology like this. So here's just a quarter of an axon, very high power EM. And here's the myelin sheath. You remember on the previous picture, the myelin was nice and dense and compact. So what these folks saw very often is that the very inner layers of myelin started to split and spread in what later turned out to be MS lesions. Now, to me, if autoimmunity and inflammatory cells are the cause, I mean, they're coming in from the outside, of course. If they're attacking myelin, the last place I would think myelin would start to degenerate would be on the inside. I would think the myelin would start to be damaged on the outside, whereas exactly the opposite is true. Moreover, these folks did not see tons of inflammatory cells floating around, eager to eat the myelin and eat the axons. So it appears that the very first problem of occurs at the very inner layers of myelin, which is very unusual. Now, when you come into the treatment world in MS, to me, more flies in the ointment. So decades of investigation and effort by researchers and drug companies have yielded very effective and powerful anti-inflammatory drugs. We have humanized antibodies, we have the extreme stem cell transplantation of the immune system to reset the immune system on the assumption that MS is caused by autoimmune. Antibodies such as Campath, Woggle, Tysabri are extremely effective at shutting down inflammatory lesions in the brains of MS patients. 90% plus reduction in lesion activity. So relapsing remitting patients that have lots of inflammation, when you measure their disease activity by inflammation, these drugs and these maneuvers are highly effective. The problem is, if you look later, the secondary progressive phase of MS is not much altered by even the most powerful maneuvers aimed at reducing autoimmunity and inflammation. And that, to me, is a major flaw. Because if MS is primarily an autoimmune disease, you reset the immune system. Shouldn't you cure the disease? No, and we don't. We don't even alter the later progressive phase very much. So that, to me, is bothersome. So there's no question to me that recurrent attacks, like I showed you on the video, are bad. I mean, that is inflammation, there's cytokines, glutamate, and all sorts of nasty stuff that is secreted by these immune cells that are rushing in. And that secondarily causes even more injury to something that might be going on underneath. So there's no question that recurrent inflammatory attacks are bad, and therefore there's no question that treating those attacks with these agents is a good thing. But that doesn't mean we're going to cure the disease, and suggestions are we are failing to do so. Doesn't mean we're not doing good for our patients. It just means we're not addressing the root cause necessarily. So then you look in the literature some more. So we're going to move away from MS, and I think when you try to crack a nut like this, you have to think pretty broadly. So I looked in the literature, and you start looking at mistakes that were made by clinicians such as myself that people have published, and from which we learn quite a bit. So I'm just going to give you, this is one of many examples that you can find in the literature, but this is a good one. So here's a case of a 49-year-old woman. She had a long history of gait problems, balance problems, slurring of speech. When her neurologist examined her, she had slurring of speech. She had trouble walking independently. Her balance was off. Her legs were stiff. Her reflexes were brisk and was suggestive of problems with the upper parts of the motor system in the brain and spinal cord, and she had sensory problems. So many of you have experience with MS, personally or otherwise. You will recognize that's a very common symptom collection that MS patients may experience. It gets better. So this patient had testing, and her visual evoked potentials, which is an electrical test that we use to measure conduction, visual pathways that is very commonly abnormal in MS patients. They were abnormal in this patient. This patient had an elevated, what we call IgG index in the spinal fluid, and that is a direct demonstration of antibodies and autoimmunity occurring within the central nervous system in response to something. All very, very common features of the MS patient. The MRI was very typical of an MS patient, including, you can't see very well, but including lesions in the cervical spinal cord, which is a very common location for plaques in MS patients. So everything pointed to a primary progressive type of diagnosis. Any one of us would have immediately made this type of diagnosis. So the patient was treated with steroids and anti-inflammatories, and as expected, showed no improvement, which these types of patients do not. But I left out a very important point in this case. Here it is. So when you dig deeper, the patient had a family history. Her son died at the age of 10 years of some bad congenital neurological disorder that wasn't identified. The pathology in the youngster showed a dysmyelinating leukodystrophy. Leukodystrophy means a problem with the white matter of the brain, and dysmyelinating means the myelin never formed properly. Not that it was normal and it got broken down. This child's myelin was never made properly. So this patient had some kind of metabolic genetic problem, it appears. And when these investigators dug more deeply, they did a genetic analysis on the son and the mother, and they found they both harbor a mutation in the PLP1 gene. PLP is proteolipid protein, it's the major protein constituent of myelin. Without PLP, things go very bad. And so the mother was heterozygous, that is, she had one normal copy. So she survived into adulthood and only later presented with the problem. Because this is an X-linked gene, the male son only had a single bad copy, so he unfortunately died at a young age. Why do I show this? Because this is a very good example of an identified metabolic genetic defect that started in the myelin and later in the mother caused a disease that looked everything like MS, but wasn't. So when I see examples like this, I wonder, well, what if MS is also such a disease, except we have not identified this problem yet? Awesome. So just food for thought. So we come back to this cartoon, and with that backdrop, makes you wonder, well, inflammation is promised, but cytodegeneration could be first. I think it's an important question. So I'm not saying I'm right. I'm saying I'm not necessarily wrong if I propose that. Okay, so let's move on. So with that question, where do we start? I mean, human diseases are extremely complex. You have to divide and conquer. You have to figure out where to start, what to look at. So I showed you the two main problems. Myelin breaks down, axons break down. So when you see that, where do you start? You can't work with humans. You have to go to animal or cellular models, or here's how we do it. So I'm just going to very quickly, I can't go into details because the time is short. Probably put you to sleep anyway, but I'm just going to give you a very quick overview of some of the techniques that we have developed over, well, 2 decades now to try to get at the nitty-gritty of what might be going wrong. So here we are. We've got myelin, we've got all the dendrocytes, the cells that make myelin in orange, and we've got the axon, nerve fiber, the copper wire in gray. So how do we interrogate these? How do we divide this up and study it? So we spent a lot of time, effort, and money, of course, developing some very precise imaging techniques to try to get at this, and here's one of them. So we use 2-photon microscopy, which is a fancy way to illuminate tissues and cells and get microscopic images. The problem with myelin is that the spacing in myelin is tiny. It's 4 to 5 nanometers. That is about 100 times lower than the optical resolution of one of the best optical microscopes. So how do you actually image what is going on in myelin, what might be going wrong? So I don't have time to tell you all the technical details, but we use some very advanced laser systems. Here's one of them. They're not just like a laser pointer, trust me. They're expensive, they're very powerful, and they offer information about biological samples that are just out of this world. So here's one, here's another titanium sapphire-based laser. So what you do, and the key is they're pulsed. So unlike this laser pointer, they emit very brief high-intensity pulses, and when that happens, very strange things happen when that light interacts with matter, and that's 2-photon microscopy. So here's an example. It's not projecting very well, but this is a living white matter tract taken out of a rat. We stained with 3 different dyes. Green is the myelin, blue is the axon, and red is a calcium reporter. So we are able to measure calcium changes both in the cells, the oligodendrocytes that make myelin, and even in the myelin itself. I don't have time to go into all the details of what this showed us, but that allowed us to identify neurotransmitter receptors on myelin. So here is one of those tracts. It's cut across, so the nerve axons are red, and all these little dots and clusters are receptors, neurotransmitter receptors that we typically see in neurons and in synapses. So no one expected to see this in the myelin. So this suggests that axons and myelin talk to each other through receptors, just like neurons talk to each other. Now we know in stroke, which is the field I come from, when these receptors are overactivated, brain dies in stroke. So we thought, well, maybe these receptors are also overactivated in the white matter, in the myelin, in other diseases like MS. So that's what we're looking at. So here's another example. So that was myelin. Here we're looking at axons. So here in turn, the axons in the spinal cord are loaded with a red dye to show you the axons. The myelin's the black stuff around it. You don't see it because we can label it. And here in this pseudo color, we also put in a dye that reports calcium levels within axons. So then we do a whole bunch of manipulations to learn what axons do in response to certain receptors and so on. And that allowed us to figure out that the axon under the myelin sheath, which was thought to be a no man's land, doing nothing, after all, it's covered by big wraps of myelin, is chock full of very interesting signaling systems and molecules. All of these you find in neurons and synapses, so this raises the very distinct possibility that if you lose control or overactivate all these things, the axons might die, just like they do in stroke, brain trauma, or spinal cord injury. So anyone who wants needs trouble sleeping, please read our papers here. Guaranteed it will have a great experience. So back to MS histopathology. So for 150 years, we've known about these swellings and things. But to this day, we have no idea what causes it in the MS brain. So we transplanted that into an in vitro model, a model under the microscope from rats or mice. And we succeeded in replicating quite accurately all the swellings, myelin breakdowns, and pathology under the microscope in real time. And I can show you movies of how this happens. It's really quite fascinating. All the different patterns of axonal swellings, myelin swellings, all the things that go wrong in the MS brain, right under the microscope in front of our eyes, which allows us to then manipulate what goes wrong and to mitigate what goes wrong, to try to learn, A, why it goes wrong, and B, find the drugs to try to stop that from happening. So this is a major effort in our lab to try to replicate the MS pathology in a dish under the microscope. So we are also geeks, and we love to push the envelope. And so two-photon microscopy is sophisticated enough. Not every lab does it, but we want to push the envelope even more. We love to live on the bleeding edge, as we call it. And so we developed a whole bunch of really neat microscopic techniques. I have physicists and engineers that work with me. Here's one example. So here's a beautiful picture of a nerve fiber in green. It's myelin in red. Because myelin is highly ordered, the dyes we put in to stain it are also highly ordered. And as a result, if you control the polarization of laser light, you've all looked at polarized sunglasses, rotated them and saw different things. So that's polarized light. Lasers are highly polarized. If you control that polarization, you can study and interrogate your nerve fibers, and particularly your myelin, in exquisite detail. So I'm happy to talk to you more about that. This is a very interesting new technique we've developed recently. So all the colors I showed you are dyes that we put into tissues and cells, and they shine in response to laser light and report things to us. But some dyes, and chemists have known this for decades, we're just catching up. Some dyes are interesting because the color that they emit depends on the solvent that we dissolve them in. So here's an example of a dye called Nile blue. If you dissolve it in a very non-polar solvent, cyclohexane, which is very much like gasoline, and shine a UV light on it, it emits green. And all the way you can do different solvents. When you start getting towards water, it gets dimmer and redder. And that's called solvatochromism, which simply says the color of light that the dye emits depends on the solvent you dissolve it in. And then I thought, well, and these label myelin beautifully. So you get beautiful myelin pictures because these dyes love lipid fat, and myelin is full of fat. So then I thought, well, myelin is the solvent. So why don't we look at the spectral signatures of these dyes when they are put into myelin, and can we learn something about the actual chemistry of myelin? And that's exactly what happened. So here's an example in collaboration with Yang's lab, where they deliberately demyelinated. This is a transverse section of the spinal cord from the mouse. So they demyelinated with a chemical, and then after about 3 weeks, the myelin recovers. And so this is a histological section of such a cord stained with this dye. And on regular, if you looked at it with your eye, you would kind of see an orange stain with a little bit of a blob over here. So, you know, a hint that something went wrong. But with our instruments, if we take at every pixel and measure the spectrum of the emission of the dyes, and then reconstruct the image and put it in pseudocolor so you can see the subtle differences of the spectral emissions better, you now come up with a very rich image. So all this blue, green, and even violet here in normal myelin that was never touched is very different, telling us the chemistry of that recovered myelin is very different than of normal myelin when it started. So this is a very powerful technique based on fluorescent spectroscopy that we are applying to fixed and also living tissue to learn more about myelination, demyelination at a chemical level. So everything I've showed you so far depends on exogenous, i.e. probes that we have to actually put in. So either you put it in from a vial, or you genetically encode it in an animal. So there's a molecule there that shines, a fluorescent molecule that shines, that emits light for your digital information. But you can also get a light from matter without any probes at all, and that's called Car's microscopy. So what this depends on, if you have not one, but 2 pulsed lasers now, so things are getting more complicated, more expensive. But if you take 2 lasers and you tune their differences of their frequencies precisely, you can get them to resonate with chemical bonds. And the bonds we chose are carbon and hydrogen. Why is that? Because CH, lipids, fats, are full of CH. So myelin, as I'll show you the next slide, lights up like a Christmas tree with So you put these two colors of light in, you resonate this bond, you get a photon out, and with a scanning system, you can actually reconstruct an image. So this is one of our earlier prototypes. So again, it's not something you buy off the shelf at Walmart and plug it in. This takes years to set up and calibrate. And I can tell you, when I moved from Ottawa to Calgary a few years ago, my engineering staff was very unhappy with me because they just set all this up, and I said, hey guys, guess where we're going? So you got to rip it all down and start over. But anyway, short-term pain for long-term gain. And it's wonderful working in Alberta. We're doing much more than we could have before, so it's all good. But this technology is really cutting edge. It's difficult, but in turn, it gives you absolutely spectacular information about your tissue. So here's an example of exquisite detail. So now we only see myelin because we're tuned to CH. So here's myelin of living nerve fibers in the dish. Beautiful detail. You can see exquisite detail, little intrusions of myelin, nodes of Brondby, incisors, and so on and so forth. So we gain tremendous information about this. And we can do this in the live animal because we don't have to put any external probes in. The CH vibration is what gives us the signal. You can also image water. You can image protein. You can image whatever you want depending on how you tune your two lasers. So very, very powerful technology. So all this, don't worry, I'm not going to go over the details of this slide. So all this has allowed us to fill in the blanks of the very complex and increasingly complex puzzle of how nerve axons work and most importantly how they die. So there are multiple biochemical and molecular pathways that we and others have identified over the last 20 years now that are bringing us much closer to our ability to identify, okay, where should we intervene to protect myelin and axons from injury? And this slide's even a bit out of date now. There's even more parts to it now. I just don't have a new version. So every month, virtually, we and others are adding to this. So here's my final point. So this is my opinion only. So that's my disclaimer. But it is my thoughts of how we might want to start looking at MS in a slightly different way than we have in the last decades. So here we are. Here's MS. The obvious thing about MS, we talked about the mushroom cloud, inflammation, autoimmune, and everything. I'm interested in the question mark, and I think there's something going on over here that caused this, so we should look here closely. So where do we start? Well, we've learned a lot about the white matter. I told you all about the receptors, et cetera, et cetera. And we know what's going on here, that we know that they're overactivated, they activate enzymes, and inflammation, and glutamate comes in, and the tissue dies. And in my mind, our view of MS has been restricted to only what's in the yellow oval. That when this happens, pieces of myelin, in my opinion in particular, are shed. And as a result, for reasons I don't know, and no one else does either for that matter, the immune system in the predisposed patient doesn't like that at all. And when that myelin is broken down and released, the immune system gets activated way more than it should and starts a cycle of inflammation, more damage, more shedding of antigens, and now you have a cyclic, propagated, chronic, persistent disease of MS. And our view has been restricted over here. We have some suggestions of what might be going on, but the key question is this one here. What started the whole thing? We don't have the answer to that. We have some ideas that we're working on. But that, to me, is the $64,000 question for the next decade. What is the original insult that got this whole thing going? And if we can block this arrow, we could perhaps stop the whole disease and not even have to worry about inflammation or autoimmunity, because then it becomes moot, because you've shut everything down over here. So that, to me, is where the money is, so to speak. So back to this cartoon. So I love this. We've actually submitted a paper with this title. I don't know how it's going to fly. But so the jury's still out. I think both are important. No question inflammation is important. No question pathology shows us degeneration. And I call it cyto. Cyto is Greek for cell, because I don't think it's neuro necessarily. Cyto is generic, it's cell. So let's keep it open. It could be any cell. I personally think it's the myelin, the oligodendrocyte, but I don't know. So both are important. The question is, what's the first step? We don't know. I think, given the arguments I presented to you, that the real MS is actually primary progression. progressive MS. Because eventually most relapsing remitting inflammatory MS turns into progressive disease. These folks start off with progressive disease. And the difference between the two is the immune system. That if you take for a moment that everyone with MS actually has primary progressive disease, the relapsing remitting patients are those unfortunate ones who, on top of everything, have an immune system that reacts to the shedding of those myelin antigens, those fragments of myelin, and now have recurrent inflammatory attacks, now have the mushroom cloud. But every MS patient, I believe, has that nuclear device at the bottom of the mushroom cloud that is brewing and ready to go on. And to me, the autoimmune inflammation, as prominent as it is, and as important as it is in this disease, is secondary. It's a reaction. You twist your ankle swells. You twist and break your ankle, you have swelling. The problem isn't the swelling, the problem is the broken bone in the ankle. We don't know what the broken bone is yet in MS, and that's what we're working on. So what implications does that have for treatment? Well, immune-targeted anti-inflammatory therapy is very effective. We all know that. It's here to stay. It's proven. It helps. So that's not going to go away. MS has the advantage, in contrast to diseases like stroke, where I work in, where if you don't get in within minutes, it's game over. MS is a chronic disease that probably started years, if not a decade, before the first attack. So when you make the diagnosis, there's still a long time for you to intervene. And so there's very favorable treatment time windows so you can intervene and mitigate the progressive degeneration that will be going on in these patients. I think the axonal, the nerve fiber, and the glia, the myelin degeneration, very prominent problem, and in my opinion, the first problem. And of course, we want to mitigate transection of nerve fibers, because once lost, as I mentioned, that's it, they don't go back. And now we have a lot of pathways that I showed you on that cartoon that we can intelligently go after with drugs, and we're working on those too. So I'm going to stop by acknowledging, of course, I don't do this. I sit in the office and write grants. It's all these very smart, talented, and dedicated students, technicians, postdocs that really work for very little money, work for very long hours with extreme dedication and enthusiasm to try to crack these problems. My collaborators from around the world, from whom I get a lot of inspiration, new ideas, and of course, the granting agencies that have supported us over the years very generously. So I think that's all I have, and thank you very much for your attention.